Welcome to the Nanovax podcast, a Nanovaccine Institute curated podcast sharing the latest research and advancements in nanovaccines and nanotherapeutics for human and animal health. The Nanovaccine Institute is a consortium of researchers, institutes, national laboratories, and companies founded and coordinated by Iowa State University.
We are developing nanovaccines and nanotherapeutics for respiratory infections, neural disorders, antimicrobial resistance, cancer, aging, and animal health.
Our vision is that nanovaccines and nanotherapeutics will revolutionize how we prevent and treat disease.
Hello, I'm Kathleen Ross.
I am Balaji Narasimhan.
I'm Mike Roof. This season, you will hear from experts across a range of disciplines who study respiratory diseases and antimicrobial resistance.
The first half of the season will focus on respiratory disease and antimicrobial research within the Nanovaccine Institute. The Nanovaccine Institute is pioneering the development of novel, nano-based countermeasures, such as a universal influenza vaccine. We will also talk about other emergent and impactful diseases that have radically changed how we consider and develop policy around global public health.
The second half will focus on the challenges and countermeasures to antimicrobial resistance. Nanovaccine Institute researchers have developed novel nanomedicine therapies to treat chronic disease caused by bacterial pathogens that have developed antimicrobial resistance.
Episodes will explore the different corners of these research topics through presentations on the latest findings as well as conversations among the experts.
Hello, everyone, and welcome to this episode. I'm Balaji Narasimhan, Director of the Nanovaccine Institute, and Anson Marston Distinguished Professor and Vlasta Klima-Balloon Faculty Chair in the Department of Chemical and Biological Engineering at Iowa State University. On today's episode, we will be talking about the immune response to bovine respiratory infections in cattle, particularly the respiratory disease called respiratory syncytial virus, or RSV. Here to discuss these issues with me today are two experts, Dr. Jodi McGill. Jodi serves as the Interim Associate Dean of Research and Graduate Studies in the College of Veterinary Medicine and the John G. Salsbury Endowed Chair and Associate Professor in Veterinary Medicine at Iowa State University. The McGill Lab, which has been at Iowa State since 2018, currently studies the response to several respiratory pathogens, including bovine RSV, and also Mannheimia hemolytica and Mycobacterium bovis, which is the causative agent of bovine and zoonotic tuberculosis. Major projects in her lab are focused on the importance of non-conventional T cells in the response to respiratory infections. She also studies novel vaccine strategies to prevent respiratory disease and investigates the impact of certain micronutrients on the host immune response and resistance to respiratory infection. Also with me is Dr. Randy Sacco. Randy is a lead scientist and research microbiologist in the Ruminant Diseases and Immunology Research Unit, or RDIRU, at the National Animal Disease Center, which is part of the USDA. The Sacco Lab, which has been in existence since 1997, has focused studies on innate and adaptive immune responses to bovine respiratory disease complex, or BRDC, viral and bacterial pathogens, including RSV. Development of viral and bacterial co-infection models that mimic this BRDC is a key part of the research effort at NADC. Scientists in the RDIRU work on innovative intervention strategies such as vaccines that would be used to reduce economic losses incurred by cattle producers. Jodi and Randy, welcome to the podcast.
Thank you, Balaji, for the opportunity.
Yeah, thanks. Thanks for that intro.
So just as we get started here, both of you are sort of in the Ames innovation ecosystem that is associated with a Research One University, also home to a lot of cross-disciplinary kinds of centers and institutes, like, for example, the Nanovaccine Institute that all three of us are part of. And then in Randy's case, he's at a federal lab, a USDA lab. So my question to both of you is, how does the presence of these entities and the presence of this sort of vibrant ecosystem, how has it helped you get to where you are in your research labs? So maybe you can speak in terms of an evolution and how this ecosystem is helping us advance novel solutions?
Yeah, so I can start. So I have kind of a long history in Iowa, I would say. So I did my graduate work with Dr. Kevin Legee, who is also part of the Nanovaccine Institute, which is where I first started learning about the center here at Iowa State. He's actually at University of Iowa. And then following my graduate work, I came to USDA and actually did a postdoc with Randy, which is how we started forming our collaborations. He was a great mentor, and then I ended up sticking with bovine immune responses and studying RSV because of the work he allowed me to do in his lab. So then I went away to Kansas and then came back because Iowa State is awesome. As you mentioned, it has a lot of really great opportunities. The vet school at Iowa State is for doing the animal work that I need to do is, awesome. It gives me all the opportunities that I need, but also the proximity to the USDA and a number of other folks that are doing animal health research in some of these agricultural species, because it's challenging. And so having access to folks who think the same and who can help you with reagents or kind of think through challenges with your experiments is a great opportunity. And then, of course, our work at the Nanovaccine Institute has been excellent. We've done a lot of vaccine work. We've done some work with innate immunomodulators, which we'll mention later, and that has really helped my research along a lot and shaped where we study.
So I started out in Iowa, then I left and went to Texas A&M and then the Ohio State University, and then made my way back to the University of Iowa for a few years. So I knew quite a few of the individuals at the University of Iowa when they joined the Nanovaccine Institute as well. Then I came to Iowa State and then moved out to the USDA, where I didn't have to write grants to have my lab funded, but it turns out I'm writing more grants recently. And as Jodi mentioned, she joined my lab, did a fantastic job there, got a number of publications in the field, and we've been collaborating ever since. As she mentioned, there's a lot of great collaboration in Ames between the university and federal laboratories. Sometimes we share some of the centers. So, we might do work with a proteomics facility or DNA facility here, that kind of thing. And so having, again, that expertise in Ames has been quite beneficial for my research. And we also sometimes have graduate students from Iowa State that will work in the USDA laboratories, conduct research, and then get their degree through Iowa State. So that interaction has been very fruitful for my laboratory also.
And I think in addition to the excellent points both of you made, now we can add the triangulated asset that is brought by industry. There's an increasing amount of animal health-related industry that's present in the central Iowa area, which is, I think, also a great addition to the ecosystem we have, and that helps us translate products even more quickly to benefit both humans and animals.
Yeah.
So maybe to get us started, how would you describe the impact of BRD on the dairy industry?
Well, it's a problem primarily in the dairy industry and calves, heifer calves, which are a critical part of that industry. But it's also very important for the beef cattle industry. It's not only in calves, but also seen in the most important cause of morbidity and mortality in feedlots. So extremely important to both industries.
I think it's important to note too that it's not just morbidity and mortality due to the infection, but it also it costs the producer a lot to vaccinate and to try to control and treat the disease. And if a calf gets a very severe infection when they're young, they almost never perform the same. The odds of them leaving the herd before they get to first lactation in the dairy or to make it to the feedlot is really reduced.
So needless to say, BRD has a very, very high impact on this industry. So in terms of that impact, how is your research helping tackle this burden on the industry?
So there are a number of vaccines currently available in the two cattle producers, but their effectiveness in the field has been questioned. In fact, some of the vaccine companies know they need to develop, more efficacious vaccines, especially because the calves oftentimes are vaccinated under stressful conditions, shoot work, weaning, shipping, that kind of thing. So it's hard to know how some of our vaccines are going to perform under stressful conditions unless we try to reproduce those in the laboratory.
Yeah, I agree. I think when we ask that calf, I mean, regardless of the state of how the vaccine is going to work, if that calf is already not at its healthiest or not ready to respond optimally, that the vaccine isn't going to work very well. So some of our research is focused on, how can you optimize that calf's immune system so that they can respond really well to the vaccine. So we have some studies focused on that, or thinking about nutrient supplementation with some of our collaborators in animal science. And, if you can get that balance right, then the vaccine might have a chance to work well.
Do you also study how to simulate those stressful conditions in your research?
We try to. In fact, sometimes, moving calves from our pasture into our confinement building kind of simulates that, stressful condition. We haven't done, there's some folks that have done transport. Actually, you put them on a truck, transport them around, and then brought them back to the lab to do their challenge. We haven't gone to that level, but if we can do some of those kind of things to try to reproduce the model in the lab, that's really a key piece that we're sometimes missing, is that it's hard to replicate those field conditions in the laboratory.
Mm-hmm.
So we've heard a lot about terms like One Health and the connectivity between animal health and human health, and the perspective that there's a lot of interplay or back and forth or learnings that can happen in a bidirectional manner. So how does your work in that context connect to human viruses?
So, before we maybe dive into specifically the virus, I think it's important to note the immune response, or the immune system, is really conserved across lots of different species. And so when we think about the response to respiratory infections in human, cattle get a lot of the same kind of challenges. So, post-COVID, everybody's a little bit of an expert on the cytokine storm and lung damage that happens when your immune response overreacts. And I think that's a lot of what happens during BRD in calves. And so understanding both the pros and cons of this robust response is helpful from a One Health perspective. From a virus aspect specifically, we study bovine RSV, which is very closely related to the human RSV virus, which causes infections in especially infants and young children, and everybody after COVID, right? And so in a lot of ways, bovine RSV models some of the things that we know about human RSV. So it tends to infect mostly very young calves. When we're trying to immunize against it, one of the challenges is dealing with maternal antibodies. So BRSV is endemic in the population. So nearly every mama cow has antibodies specific to RSV, which helps protect the calf, but also prevents some of our vaccines from working their best, which is what we also encounter in humans. And some of the immune responses and changes that happen in the lung when that virus infects is very similar between the human infant and the neonatal calf especially. So those are all kind of things we can learn from both directions.
I think it's interesting to look at a lot of different models as we're thinking about developing vaccines for humans, because we can't test those vaccines oftentimes for the first time in humans, so we have to start out with sometimes laboratory animals like rodents, and then maybe go to cattle before we think about going into humans. But there's a lot of differences. Mice aren't naturally susceptible to RSV. Their age dependency is not the same as it is for cattle. So there's multiple things like that. The upper respiratory tract of mice doesn't have the same lymphoid structure as humans, whereas cattle more closely matches that. So there are several things. but all models are models, and so they all have their advantages and disadvantages. And so we need to study multiple model systems before we actually try to think about going into humans. And that's where we've been involved sometimes with individuals that come to us with a new vaccine that works in mice and cotton rats in the case of RSV. Can we use it to protect calves? And if so, then maybe it would be a good vaccine to begin human clinical trials. So that's how we can kind of also fit in sometimes.
So can you talk a little bit about the couple of new RSV vaccines that have been approved recently by the FDA for use in humans in terms of what they're useful for and what limitations do they have?
Sure. So to a certain extent, I'm certainly not a human doctor. But so we know that the new vaccines that are out target the pre-fusion form of the F protein. So the F protein is on the surface of the virus. And for a long time, people have studied the F protein, but it wasn't until maybe 10 or 15 years ago we understood that that protein kind of goes through a change when it binds onto cells in the body. And so the version of that protein that we targeted for a long time wasn't really very good at inducing immune responses, whereas now we know that if we target that pre-F form, that's actually the response that our body wants to recognize, and it has very potent neutralizing activity. So a lot of, the currently available vaccines are targeting that pre-F vaccine, which is why they're able to get such a better response than some of the prior vaccines that have been explored.
Yeah, in fact, one of those vaccines was tested in cattle as a model before they went into human clinical trials. So again, it kind of fits with some of the things that we're trying to work on in terms of testing these vaccines in a calf model before we move into human clinical trials.
So as a follow-up to that, I know both of your labs are also interested in studying next-generation vaccines against respiratory diseases such as RSV. So could you comment a little bit on work that your lab has performed with respect to some of those next-generation vaccines, particularly in the context of RSV?
Yeah, so Randy and I collaborate on this project along with you, with folks at the Nanovax Institute. So one of the issues with the pre-F vaccine is while it's very potent and induces good responses, it's very unstable. And so they've figured out some ways to form different chemical bonds between the protein to make it a little bit more stable, but it's still expensive and hard to make. And while that's good for humans, it's probably more challenging for cost-wise in animal studies, especially. But that unstable form of the pre-F makes it hard to use as a vaccine antigen, right? It's hard to store, it's hard to make. And one thing about the nanoparticle-based vaccines is that it can protect the antigenic payloads. And so it can help protect those unstable proteins so that they last longer. It's also able to increase the potency, so it takes less antigen to mount or to induce an equally strong response. And so those are some really nice things about the nanoparticle delivery system. And so we've been taking advantage of that, and we've done some calf work recently with the pre-F protein, using it both delivered intranasally, so kind of activating immunity in the respiratory tract, as well as parenterally, so injecting it so you're going to get systemic immunity. Hopefully having both of those responses to this pre-F protein would be very protective for young humans and calves.
In addition to that, my lab has also done some work with a Newcastle disease virus vaccine, which is already approved for use in poultry. And some collaborators, Mount Sinai, inserted the RSV fusion protein into that virus. That virus induces a really good type 1 interferon response, which is critical for an antiviral response, and RSV doesn't do a good job of inducing type 1 interferon. So we have also studied that vaccine and published a paper in the last few years on the efficacy of that Newcastle disease vaccine containing RSV fusion protein.
So just as a follow-up to these next-generation particle-based vaccines, so what might be some advantages with respect to delivery, deployment, and distribution of these types of next-gen vaccines in a livestock setting?
Well, a couple things that come to mind would be delivering of one dose and needle-free. Now, also, the temperature sensitivity, or not, it's not necessary that the vaccines are refrigerated. It'd be some things that would come to mind there.
You also mentioned, one of the exciting things going forward is multivalent vaccines. These next-gen vaccines could potentially be also be used to deliver, countermeasures against multiple threats, right?
Mm-hmm. Yeah. I mean, you could encapsulate or cocktail lots of different agents together, which I think there are multivalent available now, but who knows how well they kind of target all of the different pathogens once they're all mixed together. And I mean, towards the one shot or one treatment vaccine, when you think about, in a human setting, you immunize one person in a room and, it's your kid, and then you send them out and they move on. But when you're working cattle, you're running thousands of animals through the chute. So if you can do it one time as opposed to having to bring them back, multiple times for boost after boost or, annual boost, then I think that can really save labor and stress on the animal. So that would be really big for the field.
Yeah, needle-free would also be good because you don't have to worry about the potential contamination between animals.
Not to mention the safety of the operators. So both of you talked a little bit about the importance of evaluating both the safety and the efficacy of these vaccines in animal models, particularly multiple animal models. So maybe can we have a short comment from each of you on how these different animal models inform our understanding not only of vaccines that protect these animals, but also extrapolate that understanding towards humans. In particular, the animal models for RSV are different from the animal models for, let's say, flu or SARS-CoV-2. So how do these different animal models, in your own research and in your experience, help inform our understanding as we move towards human biology?
So mouse, maybe I'll start with the mouse model. The mouse model is awesome for analyzing mechanisms of protection and for some of those early vaccine development projects, because every mouse is the same, they're clonal, and so you can get a really good picture of what the response is really doing, and the tools available in the mouse model to really dissect the immune response are unbeatable. However, studying responses in neonates in a mouse model is pretty challenging because they grow so fast, right? They're not neonates for very long. And they often don't have that pre-exposure like a human infant. So they don't have maternal antibody, or they maybe haven't seen RSV, which is what makes them good, but also challenging to extrapolate to humans. So in our model, in the calf model, we have maternal antibody. So we can test, if that vaccine is given to an infant who already has circulating RSV antibodies, how does it respond? We also have outbred genetics in the calf model versus the mouse, which is, it might work in one single population of mice that are all the same, but if you put it into an outbred population, how well does that vaccine perform? And I think those comparisons are what allow us to say, this has promise in the calf and might be worth moving into the human versus, it works in one mouse, but maybe not in a population.
And the other thing that we've done is some, safety studies, because for RSV especially, it's critical because in the 60s there was a vaccine failure where some of the infants that got the vaccine had worse disease when they were exposed to the virus itself, and a couple of the infants died. So that's been a really important thing to look at. And so we always, when we're testing our vaccine, the one thing you want to make sure of is that you don't get any untoward reactions to the vaccine, that you don't make the disease worse when you challenge with the virus. And so we can test that in the calf model, and we've done even work with colostrum-deprived calves, which are more susceptible than a conventional reared calf. And so then if it doesn't cause untoward problems in that colostrum-deprived calf, you can feel fairly confident that it's, not going to have any safety concerns. So that's one of the other places where these animal models are of benefit.
So, the world we live in, we're all exposed to a lot of different things, right? So, how important is it to study the efficacy of these next generations, both safety and efficacy, actually, of these next generation vaccines in pre-immune models, which somewhat simulate the fact that we humans are not naive to a lot of things that we're getting vaccinated against? Are there efforts to study that in the RSV vaccine field?
I think there are. I think they're still really in the very early stages. I think that will be a really exciting field to follow over the next 10 years as we start to understand how, whether it's RSV or flu or COVID or, how does the immune system change based on exposure to some of these other agents? And I don't know that we have even the inkling of how it really works yet.
Yeah, those things are really difficult. In fact, if you go back to bovine respiratory disease complex, which is bacterial and viral infections, oftentimes combined, we still are trying to learn about how do these viruses predispose to secondary bacterial infections, and each virus is different. And so you have to study each virus separately as how it's modulating the host response to allow them to be susceptible to these secondary infections. And so that's another consideration that we have been trying to look at.
So you mentioned flu and SARS-CoV-2 and some other viruses. So the data is showing that these types of respiratory viruses disproportionately affect people who are either very young and people who are that on the other end of the age curve, if you will. Is that something that's also observed with RSV? And I know that the FDA-approved vaccine currently is, I believe, for people over the age of 60. And so are there efforts to also address the enhanced susceptibility that young children face with respect to RSV in terms of the vaccine fields?
Definitely. So for RSV specifically, the other vaccine that's been approved is actually for maternal immunization. So if you immunize mom, then you can protect baby at least through those early couple of months when they're most susceptible. I think eventually they will also try to develop a vaccine that can be used, starting at the first year of life or whatever that might be. But I think they're not there at that point yet. However, there have also, in parallel to vaccine development, there have been a lot of really good advancements in developing monoclonal antibodies. And so maybe Randy can help me with the name, but I know there's also a new licensed monoclonal antibody that's much more affordable and has shown really great help in protecting preterm infants. Do you remember the name of that? I don't know.
Sorry,
It's going to be a quiz.
Yeah, exactly. I don't. Speaking of monoclonals, there's some good ones now, for SARS-CoV-2, and there's also some individuals developing nanobodies in camelid species. They're heavy chain only antibodies that have been shown to be effective against things like SARS-CoV-2. So those, a lot of companies and a lot of scientists are involved in, trying to develop some other treatment modalities in addition to the vaccines.
Well, it sounds like with studying pre-immunity, with studying vaccines that are age-specific, especially for infants, and the discovery of novel biomolecules that help augment the countermeasure portfolio in addition to vaccines are all exciting developments that are happening in general in the field of countermeasures against respiratory infections. But just to finish off, what is most exciting to you in terms of what's happening in your lab and what's happening in the field with respect to RSV that you're enthused about pursuing in the next, let's say, 5 to 10 years?
So in our lab, we've been so we kind of started working on nanoparticle-based vaccines that target the adaptive immune response, and so that's inducing T cells and antibodies, which are great. However, they're very specific, right? So if you are targeting RSV, they work great. But if you accidentally have a PIV infection instead of RSV, that vaccine is not going to be protective, right? So the project that we've been working on kind of recently is more about innate immunomodulators. So if you can, this kind of goes back to setting up the immune system to work its best, right? So if you can activate the innate immune system to be poised to protect that calf or that infant from whatever they might see, be it, a virus or a bacteria, kind of during these periods where we know that they're at high risk, that might be able to fend off disease long enough for some of those vaccines to get on board and to work better. So we're really excited about studying innate immunomodulators and then also thinking about how treatment with those can help vaccines work better in the long run. So that crosstalk between innate and adaptive immunity.
I think it's exciting that we finally have a couple of vaccines in the market that are approved for use against RSV, because all kids have been exposed by the time they're 3 years old. It's a continuing problem. Where I also think we need to have some effort in the next several years is in developing countries. It's still, RSV is a real problem worldwide, especially in developing countries. They don't have access sometimes to the same medical care we do. The monoclonal antibody that's approved for use in treating at-risk infants is very expensive, and if you don't have health insurance, you probably can't afford it. It's like $1,500 a dose for 6 doses, and so you can imagine that a lot of people without health insurance can't afford that. So I think if we really need to try to figure out how we can deliver some of these to developing countries. And so here's where a nano vaccine might have an advantage, where the cold storage kind of thing isn't a critical issue. So I think that's where I hope we can concentrate some of our effort in getting vaccines that are affordable in some of these countries, made available to people. Great, we have them in the U.S., but I'd like to see them, available for others. And then in terms of also trying to deliver new vaccines to cattle producers, which is one of our, charges with USDA, National Animal Disease Center. Figuring out how to, a lot of times those vaccines are multivalent, so multiple viruses, multiple bacteria. So figuring out how to get that delivered in a nanoparticle would be, an interesting approach as well.
Wow. Yep. I'm very excited listening to these ideas about what's coming, and I love the vaccines for all kind of concept where we can get these vaccines to everyone, both human and animal. So thank you again, Jodi and Randy, for a wonderful simulating discussion, and thank you everyone for joining us for this discussion. Other episodes this season will explore other facets of respiratory disease, both in livestock and humans. Please check out the show notes to learn more about the research discussed in this episode. And please be sure to like and subscribe to the Nanovax podcast in your preferred app or media player.